Mendel's work with the peas was absolutely groundbreaking, but not all traits show those same patterns of inheritance. For most traits in most organisms, genetics is a bit more complex. For example, many traits are controlled by more than one gene, and many genes have more than just two alleles. So this week, we're going to learn about those other patterns of inheritance. First one is incomplete dominance.
All right, so first one we're going to learn about is incomplete dominance. And this is where neither allele is dominant or recessive. So for our first example of this, we're going to look at these 4 o'clock flowers is what these are called. So what are we going to find when we cross a red one with a white one? Let's see. The offspring are pink. So let's think about the genotypes here. If we use big R for red, this would be big R, little r for white, little r. Our offspring is heterozygous. And so in incomplete dominance, if they're heterozygous, since neither allele is dominant or recessive, they mix. And so we get this blend of pink from a red and white flower. So if we put this into a Punnett square real quick, we can see we would use the Punnett square just like before. We take one parent's alleles and we split them up on one side of the Punnett square because of meiosis. We take the other parent's alleles and split them up here. And in all of these, all of their offspring would be heterozygous, and all of their offspring would be pink because this trait shows incomplete dominance.
Now, one example of incomplete dominance that we can see in humans is a disease called familial hypercholesterolemia. It's a long word, but, well, it's 2 words. But in this disease, your body is not able to remove LDL, or the bad cholesterol, from your blood. And so there are 2 alleles in this case. One is for making liver cells that have these LDL receptors. The other is for making liver cells that don't have these LDL receptors. So if you get 2 alleles that make the receptors, then your body is able to remove that cholesterol from your blood. If you have 2 alleles that code for not making those receptors, then your body is not removing that cholesterol from your blood, and you would have really high cholesterol, extremely. But if you have one of each allele, if you're heterozygous, then some receptors are going to be made. So your body would be able to remove some cholesterol from your blood. So you would have high levels of cholesterol still, but not as high as if you had none. So it's as if you had no receptors. So it's that in-between phenotype that we see in incomplete dominance. Second one is codominance.
All right, so codominance. This is a situation where both alleles are dominant. And so let's look at a different flower example. Let's look at a rhododendron. This is another type of flower. And if we cross a pinkish, reddish rhododendron with a white one, we actually get one that has pink and white. So they're not mixed. It's not like light pink. We see both because both are dominant. So in cases like this, we actually use 2 different letters now because both are dominant. So let's use reds, R's for the red. I guess it's kind of pink, but we'll use R's. And then W for the white. So this would be each of the parent genotypes. And then if we do our Punnett square to see the genotype of the offspring there that shows pink and white, or red and white, that offspring would be RW. All these offspring are. That's the only offspring that can be produced from this cross because the one parent can only give an R, one parent can only give a W. So we use two different capital letters because both are dominant. So we could still call this heterozygous because the two alleles are different. And so we're going to see both phenotypes. Not blended, we see them both completely.
Human blood types are an example of codominance, because if you get an allele for type A blood from one of your parents and an allele for type B blood from the other parent, then you'll have type AB blood. And so it's where we see both of those expressed. Codominance. Third, we have multiple alleles.
All right, so our next one is multiple alleles. Now this is when more than two variations of a gene are present, because remember that alleles are the different forms of a gene. So our example for this is actually going to be blood type still. And for blood type, there are 3 different alleles that you can have rather than 2. So the first allele, we write them on in I for blood type. So IA is the A allele. There's also the B allele. And we write them like that because remember from codominance, both of these are dominant. And then there's a recessive allele. So we use a lowercase I, and that one, if you had 2 copies of that, would give you type O blood. So if we look then at this chart, then this shows us the different genotypes that you can have and what blood type that would give you, okay? You can't see down here, but this one says this one's AB, and this one is type O blood. So that's multiple alleles because rather than just having 2 different forms, there's 3 different alleles.
Last but not least, polygenic traits.
Now, polygenic traits. Before I put the definition up there, we can get some clues because poly means many. Polygenic, so many genes. And these are traits that are influenced by more than one gene, okay? So many genes influence these. And so, for example, height, skin color, eye color, these are all examples of traits that we have that are controlled by more than one gene. And so that's why they're called polygenic traits.